Today's lesson is on active and bulk transport. We've been talking about cell transport, and we've already gone through the different types of passive transport, which is simple diffusion, facilitated diffusion, and osmosis. Today we're going to talk about active transport and bulk transport. With active transport, the materials move across the cell membrane with the use of cellular energy. So this is opposite from passive transport that requires no energy. Active transport requires energy. The molecules are transported against a concentration gradient, so it goes from an area of low concentration to an area of high concentration. This is opposite from passive transport, which goes along a concentration gradient from an area of high to low. Whereas with active transport, you need energy because you're going against that flow. So you're going from an area of low concentration to high concentration. So you're going to need that energy to go against that gradient. Energy for the process is powered by ATP, which stands for adenosine triphosphate. Energy is released when the phosphate bonds are broken, and ATP becomes ADP, which is adenosine diphosphate. You can see that in the image where it goes from ATP to ADP. ATP provides all the energy that's required by the cell to power activities like muscle contraction, locomotion, and cell division. With active transport, materials go from an area of low concentration to an area of high concentration against a concentration gradient using carrier proteins and ATP. You can pause the video to make notes of these diagrams. This here just shows how a solid can go from inside of the cell to out of the cell, and how a solid can go from out of the cell to inside of the cell. With active transport, nutrients from digested food need to be absorbed through the small intestines. If there was only passive transport that was used within the cell, then only half the glucose molecules could actually be absorbed. The image below just shows an example of sodium and potassium pumps that are used for nerve and cell function that require active transport. And that is active transport. So now we're going to move on to bulk transport. With bulk transport, there's two types, and the easiest way to remember what bulk transport is if you look at the word bulk. Bulk means kind of like heavy, large quantities. Think of bulk barn or even buying things from Costco. Things are bought in bulk. So with bulk transport, large quantities of materials are moved into or out of the cell. And there are two types of bulk transport, endocytosis and exocytosis. Endocytosis has two types, which are phagocytosis and pinocytosis. So with endocytosis, large amounts are brought into the cell from the extracellular fluid, and there are two types. There's cell eating and cell drinking. Phagocytosis stands for cell eating, and that is a transport of solids into the cell. What happens is there are these finger-like projections called pseudopods that reach out and enclose around the molecules, forming a vesicle in the cell. The lysosomes then fuse with the vesicle to break down the contents. Whereas pinocytosis is cell drinking, and that would be the transport of liquids into the cell. If you're looking at this image, which one do you think it is? Phagocytosis or pinocytosis? If you guessed phagocytosis, then you are correct, because phagocytosis would be cell eating, so it's kind of the movement of solids into the cell. Next image, phagocytosis or pinocytosis. It's a 50-50. If you look at the image, you'll notice that all of those little dissolved solids would represent the liquid. So this here would be pinocytosis. And you can even see in the label there where it says pinocytotic vesicle, gives you a hint that it is pinocytosis, so cell drinking, bringing liquids into the cell. And then now moving on to exocytosis. The last type of bulk transport is exocytosis, and that is the movement of large amounts of material out of the cell through secretory vesicles. You can pause the video if you want to draw that diagram into your notes. What you'll notice is that the proteins first enter the endoplasmic reticulum as they're made by the ribosomes. Then the protein travels to the Golgi apparatus, through the Golgi apparatus. Then the secretory vesicles containing the protein forms at the end of the Golgi apparatus where it's about to be pinched off. Once it pinches off, it's in the secretory vesicle, which then attaches to the cell membrane, and then the protein is secreted into the extracellular fluid. and that is exocytosis. And that wraps up our cell transport lesson. We have now gone through the 3 types of transport: passive transport, active transport, and bulk transport.